Hello. Today I'll be presenting my proposed work on the development of a detection test that can both detect and differentiate between the SARS-CoV-2 variants of concern using CRISPR technology. My name is Gabriel Lamothe. I'm a master's student under the tutelage of Professor Jacques P. Tremblay. This work is being performed under the Faculty of Medicine in the Department of Molecular Medicine at Laval University. Studies have confirmed that many individuals infected with the SARS-CoV-2 virus are asymptomatic carriers of the virus. This is problematic in that the pandemic might be growing without adequate supervision. Simply focusing the testing on individuals with symptoms as opposed to entire populations is not enough. To better contain the virus until a large-scale distribution of an effective vaccine is underway, large-scale and continuous testing is mandatory. The current diagnostic pipeline for detecting SARS-CoV-2 is a three-step process. The first step is to obtain nasopharyngeal swabs or gargrism samples from patients. The next step is to isolate the total RNA from said samples. The RNA is then used in a real-time quantitative PCR machine. The RNA is first reverse transcribed into DNA and then amplified. A dye is added to the amplification reaction to signal the presence of DNA strands. After a certain number of amplification cycles, a positive result will give a strong enough signal that the machine flags it as containing the original virus. Now, this process is problematic for a few reasons. The first is that real-time PCR machines are extremely expensive. Most range from $15,000 American dollars to over $90,000. Given that it's a fairly specialized piece of equipment and that very few molecular biologies actually need one, most get along just fine with a regular PCR machine that costs a much more reasonable $5,000. As such, widespread testing for the coronavirus using these machines isn't ideal. Acquiring the large number of machines required for widespread testing is a feat in and of itself and is likely to be one of the contributing factors for the sometimes long wait times to get results. Many reports have stated that it can take 3 to 24 hours or more to obtain results in a clinical diagnostic laboratory. Over here in Canada, some people experience wait times of up to 4 days to get results, and high-density metropolitan areas are even more subject to long lines for testing and can have a time to results of 5 to 7 days in some clinics. The creation of new types of detection tests is therefore an important step in curbing the spread of this virus. With our previous test, which focused solely on detecting the presence of the SARS-CoV-2 virus, but not differentiating between the variants, we had an excellent sensitivity and specificity. Essentially, when the virus was present, even when the patient had too little of the virus to be infectious, we could detect it. Now, this test is not as sensitive as the current gold standard, that is, RT-qPCR, which we mentioned previously. However, it can be administered in well under 2 hours and with very basic molecular biology tools that do not require any specialized or expensive equipment. We have demonstrated that we can detect SARS-CoV-2 sequences at a concentration of 10 synthetic viral RNA copies per microliter, the equivalent to less than 0.01 copies per milliliter. We also demonstrated that negative samples display negative results when using our test. While this might sound obvious, some tests give significant false positives which can interfere with proper safety measures, and can cause many people to enter confinement, even if they aren't infectious. Very briefly, the way this technology works is by amplifying and detecting the COVID sequences in two reactions. The first reaction, or the LAMP reaction, will first take the genetic material of the virus and amplify it in repeating sequences using a technology called LAMP. These sequences are then added to the second reaction to make DNA and RNA. The DNA and RNA are then useful for activating either the Cas13 or the Cas12 proteins. In this test, we've designed it such that either the Cas13 or the Cas12 is activated, as you can see here, but never both at the same time. This means that when the Cas13 is activated, we'll get a green signal, and when the Cas12 is activated, we'll get a yellow signal, but never both at the same time. From a hands-on perspective, the way this test works is very simple. Patients will first be asked to gargle 5 milliliters of water to be able to obtain the virus in an actual sample. That or nasopharyngeal samples can be taken. However, since gargle samples are generally less invasive, we'll probably prefer to use this on a wider scale. The samples will then be heat inactivated so that the genetic material of the virus is present and technicians don't run the risk of getting infected. The samples will then be transferred to the first plate, the amplification plate, and incubated at 64 degrees Celsius for 47 minutes. After that short incubation time, they'll be transferred to another plate using multi-channel pipettes, which can be done very rapidly. They'll be incubated again, and then placed under a UV light to visualize all of the different signals. I'll explain what the signals mean now. The interpretation of results for this test is pretty straightforward. Every patient is assigned 2 wells in a 96-well plate. The samples will only be fluorescent in cases where the patient is infected with the SARS-CoV-2 virus. Depending on the color combinations present in their wells, we can determine which variant is infecting the patient. A green-green combination means that the patient is being infected by the London variant. A yellow-yellow combination means that the patient has the South African variant. A blue, or negative, and green combination means that the patient has the Brazilian variant. A blue and yellow combination means that the patient is being infected with a more generic variant of the basic SARS-CoV-2 virus. As the pandemic progresses and new variants are discovered, which are more resistant to the current vaccines, we will be able to easily modify the test by changing out a few basic components to focus our efforts on detecting them and assigning them a color combination. This will be nearly as simple as changing the tires on a truck, really. When both wells are negative, as you can see here, then the patient does not have a detectable infection by the SARS-CoV-2 virus. However, this does not guarantee that they don't have an infection at all. Rather, it means that if they do, it's below the limit of detection of the test. Remember, other reports have shown that rather than having an extremely sensitive test, a simple test that can be administered rapidly, even if it has a slightly weaker limit of detection, is better for stopping the spread of the virus. Our test accomplishes this perfectly, and not only that, we can differentiate between the variants. This plate will also be fairly easy to read for the technicians performing the diagnostic test. However, the option will exist to have it analyzed on a server after taking a quick picture and uploading it. This will allow the samples to be read more quickly and accurately, as well as providing other data-driven services. For this test to work, a few common laboratory tools are needed. First off, 2 heating stations will be required. These stations can take many forms, and I'll get into that later. But basically, so long as the heating stations can provide a stable heating at 64 degrees Celsius and at 37 degrees Celsius, then they will work just fine. 2 pipettes will be necessary to transfer fluids between the steps in the detection test. To make the test quicker and simpler to perform for the technicians, one of the pipettes can be a multi-channel tool to manipulate many samples at a time, as you can see on the left. To visualize the signals, a common laboratory tool called a UV transilluminator can be used. This machine simply emits UV light evenly on a surface. We can use this to illuminate every well evenly and properly determine which wells demonstrate yellow or green signals as applicable. As you can see here, all of the tools I have just mentioned are present in pretty much every molecular biology laboratory, as well as in many others. Assembling these tools will be very simple in most settings. Additional tools can be added to simplify and streamline the entire process. When using the server to interpret the results and report positive cases, a small tower can help to simplify picture taking. A generic cell phone can also be used to take the pictures and subsequently upload them to a server so they can generate data and downstream anonymized reports. Various machines exist that can complete the incubation steps of the detection test. Essentially, so long as the machines are capable of providing a stable heating platform, they can work and provide us with diagnostic results. This means that incubators and heating blocks that are already in molecular biology laboratories can be readily used in this setting. Even old PCR thermocyclers that can't really cycle well between temperatures can be used well in this test, so long as they evenly heat all of the samples. This means that even old laboratory equipment can be recycled and used to diagnose the SARS-CoV-2 variants with our technology. Unlike the extremely expensive real-time thermocyclers that are used in RT-qPCR, which can cost around $80,000 American, and are an absolute necessity for the current gold standard in SARS-CoV-2 diagnosis. The detection test works by activating one of 2 signals when the SARS-CoV-2 sequences are present, all based on which mutations are present in the sample's genetic material. These samples are easy to differentiate because they emit different light wavelengths, which can be visualized as either green or yellow. The fluorescent readout of the test can be visualized on a common gel UV transilluminator. This is the kind of tool that pretty much every molecular biology laboratory has, similar to how an office almost always has a printer. Obtaining a few for the purposes of this test will be simple in almost every setting. Now, the results can be read directly on the transilluminator by the technicians. Really, all that they would have to do is to compare the light combinations with a paper stating what each variant could be. However, another option exists. That is, to take a picture of the plate and upload it to a server for interpretation that is free from potential human error and for data storage. For this, we have designed a simple tower that can be 3D printed on most personal and commercial 3D printers. This tower will help to block out ambient lighting, allowing the technicians to work in a well-lit environment and generally makes things much easier for them. It also helps to standardize the distances between the smartphones and the plates, as well as the angle between the phone and the plates. Here you can see a depiction of how the test can be visualized once it's completed. Simply by placing the 96-well plate on the UV transilluminator, it is possible to determine which samples have the variants. The color combinations are easy to differentiate by the naked eye and can be captured on a cell phone very easily using a 3D printed tower. The images taken on the cell phone can then be uploaded to the server using a simple app to make the diagnosis much quicker for technicians and to help inform patients which ones need to enter confinement immediately. This will also help to create anonymized reports to help decision makers down the line. When using the server to interpret the data from clinics, the entire process will be done in a few short steps. The technicians will first take a picture of the patient samples and upload the images to the server. There, the server will analyze the images to determine which wells correspond to patients that have a SARS-CoV-2 infection, and whether or not that infection represents one of the variants of concern. All of this will be done in a matter of seconds, and this data, as well as the original image, will be kept on a secured, cloud-based server. The data will immediately be sent back to the technicians, so that the patients that just gave samples will find out whether or not they have an infection in under 2 hours. The reason we have chosen to make this system work using Androids and iPhones is that these devices are extremely common. They aren't even laboratory equipment, and, well, pretty much everyone has one. In addition to providing the technicians with immediate diagnostic results, it will be possible to use the data generated to report positive cases in a contact tracing pipeline to help health advisors to better curb the spread of the pathogen. Plus, the server will be able to create simple, anonymized reports to better identify how the variants and the virus itself are spreading on many fronts. These reports can take many forms. For one, the data accumulated over the past days, weeks, or months can be interpreted to create graphs that demonstrate the growth or decline of the variants over time. It will also be possible to determine if the variants are afflicting certain geographies or populations more heavily than others by visualizing where outbreaks are currently occurring. These reports will be made automatically on the server, generating usable graphics from raw data without the need for extra personnel. This will help to reduce the cost of generating this data. These reports will also help to inform decision makers about the spread of the variants between individuals and communities and help them to make new policies to address the outbreaks and adequately handle them. Thank you for your attention as we have attempted to demonstrate the technology that we would like to create to better detect and differentiate between the variants of concern and hopefully end this pandemic sooner.